Hi, this is René Overmeer from Hubrecht Organoids, and today I want to talk about high throughput screening using organoids. So why organoids? Well, if you look at the classical 2D screening in mouse models used in a lab, these actually have very poor correlation with the clinical response. The ability to culture adult stem cells in the lab has enabled us to establish cultures from patient biopsies in an efficient manner. Being that we can establish organoid cultures from nearly all patient biopsies or patients, and that these cultures then also represent the tumor heterogeneity much better, as there's very little selection of the cell type being grown. This leads to the most patient-relevant model available. In addition, it's been shown that patient-derived organoids correlate quite well with the patient response in the clinic, and we can test combination therapies on these organoids as well, meaning that we can use them for preclinical identification of clinical response. Furthermore, we can use them as companion diagnostics, or even also do a large amount of characterization of the organoids, meaning we can use them for biomarker discovery, in the end leading to a better patient stratification to treatment and personalized care. In the lab, in the clinic now, we already have the first organoid predictive diagnostic use for the justification of reimbursement of cystic fibrosis medication, and we have multiple clinical trials based on organoids going, and also drugs developed using the organoid technology in clinical trial at the moment. So that's all good, but we also need to do some screens with them, experiments with them. So let's talk about viability screening. First of all, I wanted to point out some clear differences between organoids and 2D cell lines. First of all, organoids require extracellular matrix. Within the lab, we actually prefer to use matrigel over BME, although many tumor organoids and other organoids can grow using BME. If there ever is a difference between the two, it's usually matrigel is better. Furthermore, there's a very strong growth factor dependence of organoids, meaning that the quality of your medium can really influence the viability and proliferation and even differentiation of your organoids. So it's really key to ensure that you have a high quality reagents and test your batches. Also, not all readouts are suitable for the complex structure of organoids, so it's key to also check that the method you're using is suitable for organoid culture. In addition, organoids do not really respond well to a single cell state, so it's important that to realize this as the viability, especially normal organoids, but also tumor organoids, can be affected adversely. This means that you should avoid oversharing the organoids, as this can lead to an unwanted selection for your cell types, and also that single cell counters are not suitable for establishing the amount of our cells you're putting into a screen. So first we wanted to go over some data over the size of organoids and how this collides and the amount of organoids put in a well. We actually seeded various amounts of cells of organoids of different sizes. And you can see for the smaller organoids, we see a nice correlation with the amount of organoids and the total signal we see after 5 days. For the largest organoids, we did not see this, and it was already saturated at the smallest amount. When looking at these graphs, it's also important to realize that we should not use conditions which were saturating the system, like for over 500 organoids, as we might miss some more mild effects of your components and the proliferation. Also, because of the growth factor dependence, we were interested in seeing how long the organoids would be able to proliferate without medium refreshment in the 3 to 4 well plate. So you can see we see the different amount of organoids here, and independent of the amount of organoids in the well, up to 7 days we could see an increase of cell number, indicating that the growth factors were stable for 7 days, and after that we saw a decrease or stabilization of the cell number, indicating that the growth factor was depleted. We also tested multiple readouts. I'm going to focus on 2 here, being Alamar blue and cell-type glow. Alamar blue is something that accumulates, the substrate accumulates over time, and you can see here that the signal increases as the incubation time increases. And cell-type glow is lyses the cells and measures ATP concentration, and you can see that this is a single point measurement. When comparing 2 different organoid lines, you can see that the IC50 is comparable when using cell-type glow, which would be expected for stars born. However, when using the Alamar blue, we saw that the short incubation time worked quite well for the P18T, and the longer incubation time shifting the IC50 to the right. Whereas on the other hand, the P26T, it would seem that the longer incubation time was more suitable than the shorter incubation time to get a comparable IC50. That said, the IC50 can really depend on the incubation time when using Alamar Blue, and that each organoid line probably requires separate optimization. So within the hub, we decided to move forward with cell-type glue. When we did the first experiment seeding organoids using a multi-drop and a 5% matagel suspension and adding 200 organoids per well, and we after measuring after 5 days, the initial experiment looked quite okay with set primes of 0.4 or higher. However, when we repeated the experiment, the set primes fluctuated wildly, being quite low and negative in some cases. Looking more into detail, we saw that the raw values of the untreated organoids here fluctuated wildly, so being of 53,000 with some conditions only 2.5 for the same organoid line a week later. So this led to us having a better look, and then we concluded that the amount of viable organoids that we were seeding really varied a lot. This is probably due to the fact that we had some issues in assessing the exact number of organoids that we had, especially small organoids were difficult to count, but also that when we had a longer handling time, the viability of the organoids decreased. So we really focused on decreasing organoid handling time and standardizing the cell counting, or the organoid counting. When we did this, we did a larger screen of 13 plates and multiple replicates, and the Z prime was consistently high in this case, always above 0.5. And if you look at the raw ATP values, you can see that also in these histograms per plate, you can see that they're also more or less comparable between the different replicates, indicating that we now have a reproducible screen. This is also seen when we compare the each single point, the treatment point. They all follow more or less the diagonal that we see here between the first and second replicate, or and the third replicate, with a couple of outliers, but these were very few considering there's over 3,000 points in this graph. When we then further went and expanded this system to 10 different tumor organoid lines, we can see that without further optimization, we could get reproducible results in these different organoid lines, although some organoid lines showed a bit more variation. However, we're still, especially with replicates, quite able to separate non-responding treatments with responding treatments or effective treatments. Also, when we looked at some patient organoid lines, so these are different organoids established from primary tumors and even one from metastasis from the same patient as the screen line is, we can see first of all that the biological replicates, being the dark and the light lines, are quite close to each other, with the patients always clustering together, and that one of the patients was sensitive to all of the treatments, and one of the patient, the one with the metastasis, that both the primary and the metastasis were resistant to all of the treatments, whereas there was one patient in this case that was resistant to most treatments, but was sensitive to the SM38, indicating that the organoids can be, in this screening paradigm, can be used to classify or stratify patients to sensitive and non-sensitive to certain treatments. So just a summary of some tips and tricks. So extracellular matrix, matagel BME. The organoids can be cultured in the suspension, and this makes seeding it a lot easier. The concentration should be between 2 and 10%, and this depends on the organoid type. When doing this, really, it's important to prevent attachment to the plate. So this means that you should not use your standard culture plates with a lysine coating or another coating, but you'll use plates with a repulsive or non-adhesive coating. And also never mix the cold matagel or BME into warm medium, as it will immediately polymerize and form flakes, and you will not get an even distribution throughout your wells. Standardization of the medium is key in considering the growth factor dependence, and especially when you want to compare results over a longer time period. Also, please check that your growth factors do not have an interaction with your therapies, especially for targeted therapies like cetuximab, which is specific for EGFR receptor, can go into direct competition with EGF, which is also in the medium. Also, consider whether your readout needs to be stable over different organoid lines and whether it's suitable for organoids, and also 2D imaging technique cannot be used in organoids. So also prevent oversharing your organoids to single cells, and keep handling time as short as possible. In the hub, we do not shy away from using multiple technicians to process an organoid line in parallel to shorten the handling time, and be aware not to create a concentration-dependent bias when counting the organoids. That's when you need more organoids, that you are counting the borderline and thereby overestimating your concentration and putting less organoids in the screen and affecting the quality of your screen. So in short, we feel that the organoids are suitable for preclinical development with the screening, but also for biomarker discovery due to characterization possibilities, and we can use them for patient stratification, and then directing the right patient to the right clinical trial, and even determining beforehand whether patients will benefit from a treatment, which can be especially interesting when treatments have a lot of side effects or are very expensive. Last slide I want to share with you is our mission statement. In essence, we're a non-profit foundation which is really focusing on the development of the organoid technology and technique. So I would like to thank you for your time, and I hope you enjoyed this presentation.